The rich, savory aroma of toasted bread or seared meat emerges from a complex dance of molecular paperclips. These tiny, invisible links orchestrate transformations far more profound than you could ever imagine in your kitchen. Every ingredient in your pantry holds a universe of chemical potential waiting to be unlocked through heat or mixing. We often overlook these microscopic components, treating them as mere building blocks in a recipe. Yet, understanding their specific roles reveals the true artistry and science behind delicious food creation. Think of these critical connections as chemical paperclips, holding together the structure of flavor and texture. These molecular bonds dictate how ingredients react, forming new compounds that delight our senses. From the crispiness of fried chicken to the softness of a perfectly baked cake, chemistry is the conductor. This intricate ballet occurs constantly, transforming raw components into culinary masterpieces right before our eyes. Learning about these hidden mechanisms empowers you to manipulate flavors and textures with incredible precision.
It allows you to move beyond simply following recipes, becoming a true architect of gastronomic experiences.
Preparing food shifts from routine to a fascinating laboratory full of delicious scientific discoveries. Consider the Maillard reaction, responsible for the incredible browning and deep flavors in cooked food. This reaction begins with simple sugars and amino acids, those tiny protein-building blocks, acting as our paperclips. When heated, the amino acids' nitrogen atoms link with the sugar's carbon atoms, initiating a complex chain reaction. These initial molecular paperclips form unstable compounds that rapidly rearrange into hundreds of new flavor molecules. Imagine a single tiny paperclip triggering an avalanche of deliciousness, creating notes of roasted coffee or toasted nuts. But that's not the only way these chemical paperclips transform ingredients. Take caramelization, a sugar-only reaction, where heat breaks down sucrose into smaller reactive fragments. These fragments then reassemble, forming complex polymers that give caramel its distinct golden color and rich taste. Each reformed bond is like another chemical paperclip, weaving a robust network of new flavor compounds. The subtle differences in temperature and time dictate which specific new molecules form, creating varying caramel notes. Interestingly, the presence of even trace amounts of acid or base can profoundly alter this entire process. Another fascinating paperclip example lies in emulsification. Like making mayonnaise or hollandaise sauce, oil and water naturally repel each other, refusing to mix into a stable, homogeneous solution. Here, an emulsifier, often a protein like lecithin from egg yolks, acts as the crucial molecular paperclip. Lecithin has both water-loving and oil-loving ends, effectively bridging the gap between the two immiscible liquids. This tiny molecule physically links the oil and water droplets, preventing them from separating into distinct layers. Wait until you hear how salt, a seemingly simple compound, can also act as a powerful chemical paperclip. Salt, sodium chloride, is far more than just a seasoning. It profoundly influences protein structure and water retention. When applied to meat, salt dissolves into ions, which then interact with muscle proteins, unraveling their tightly bound structures. These ions act as paperclips, allowing proteins to relink in new ways, trapping moisture and tenderizing the meat. This process, known as brining, dramatically improves succulence and flavor by altering the meat's molecular architecture. Without these precise ionic paperclips, the texture and juiciness of countless dishes would be completely different. Consider fermentation, where microorganisms employ their own enzymatic paperclips to transform food. Yeast, for instance, uses enzymes to break down sugars into alcohol and carbon dioxide, giving bread its rise and beer its buzz. Lactic acid bacteria convert sugars into lactic acid, which preserves vegetables and creates the tangy flavor of yogurt. Each enzyme acts as a highly specific paperclip, catalyzing a particular chemical reaction with incredible efficiency. These microbial paperclips are essential for countless fermented foods we enjoy, from cheese to kimchi. And then something unexpected happened. Even the simplest temperature changes can serve as a powerful chemical paperclip. Heating an egg causes its proteins to unfold and then relink, creating a solid, irreversible structure. This denaturation and coagulation is a fundamental chemical transformation, turning liquid into a firm, edible form. Understanding these molecular paperclips empowers you to cook with greater intention and achieve consistent results. You can precisely control browning by manipulating temperature and surface moisture, optimizing Maillard reactions. Knowing about emulsifiers allows you to rescue a broken sauce or innovate new creamy textures in dishes. Recognizing the role of salt in protein tenderization transforms your approach to seasoning and brining meats. This scientific perspective helps you troubleshoot culinary challenges and adapt recipes with newfound confidence. It means you can intentionally enhance flavors, textures, and aromas, elevating everyday meals to extraordinary experiences. Professional chefs leverage this knowledge to create signature dishes, pushing the boundaries of culinary art. They meticulously control conditions, understanding that small chemical adjustments yield significant gastronomic impacts. Home cooks can also apply these principles, transforming their kitchens into laboratories of delicious experimentation. Imagine crafting the perfect crispy crust or the silkiest sauce, all thanks to your understanding of molecular bonds. This insight into food science provides a deeper appreciation for the complex processes happening in every bite. It unlocks a new level of creativity, allowing you to innovate and personalize dishes with scientific precision. The world of food chemistry is constantly evolving, with scientists discovering new paperclip interactions regularly. This ongoing exploration promises even more exciting culinary breakthroughs and delicious possibilities for the future. Every meal becomes an opportunity to marvel at the incredible, invisible chemistry at play. Ultimately, recognizing these tiny, powerful chemical paperclips transforms cooking from a chore into an endless adventure. Embrace the science in your kitchen, understanding that every ingredient holds a universe of flavor waiting to be unleashed. If you found this fascinating, subscribe for more incredible discoveries and share your thoughts in the comments.